all biology students. I am Sapna Trivedi. Students, we have discussed about the cell, unit of life, where we have discussed about prokaryotic cells, eukaryotic cell structure. Eukaryotic cells just we have started and we done about the prokaryotic cells with the help of the mycoplasma bacterial cells. And I hope you are understand this topic. This eukaryotic cells we are study the step by step. Okay, so there is a division of cytoplasm, plasma membrane and cell organelles. So very important topic which we have discussed in early video also, that is the plasma membrane. So first I want to just revise the fluid mosaic model. We have studied the other model also, that is called the sandwich model, unit membrane model, and who proposed one, there is a scientist name. Okay, that is a brief explanation only we want. But detailed explanation we want from this fluid mosaic model. Okay, so let me revise this structure and then after we proceeding of function of plasma membrane. Okay student, let's see. See, you can be see this structure, and this structure is showing a fluid mosaic model which given by Singer and Nicholson. Okay, we studied its plasma membrane through the RBC cells, and there is a composition of protein, lipids, protein and oligosaccharides and water. Okay, so there is specific percentage of lipid 20 to 79%. You can note down 20 to 70% is a protein. Oligosaccharide, this is the sugar one, is a 1.5%. Water is a 20%. Okay student, let's see. As we are saying, this fluid mosaic model is showing a iceberg model. Ice we are say this is a lipids, and birds iceberg. Sorry, this is this lipids which is showing the head and tail is showing as a sea, water, sea. And here is a merging of the protein. This is a merging protein. Is just show the ice one. So we are saying this is a resemble of the iceberg one also. See, this is a summarization of this structure. We are finding this is having a phospholipid bilayer. This is a one layer and this is another layer. That's also called the two layer, bilayer. And this is a phospholipid. Okay, phospholipids which having a two region. One is a head and another is a tail. Okay, head part is called a phospholipid molecule, fatty acid of hydrophobic, which is inside in the membrane. Clear? And this part is having, this is a head part. This part is a phospholipids of such a lipids one which is having a hydrophilic. Okay, philic, philic. Okay, so this is a hydrophilic. So we are seeing in this structure you will be see there is this protein which is totally abundant. We are say this is the internal, or we are say there is an intrinsic protein. Okay, some other protein is just be on a surface, so it's called a peripheral protein. Okay, so this is also called extrinsic protein. So this is having the types of the protein which is just completely abundant, intrinsic, some part is out also. Some are just be above, that is called the peripheral protein. In this membrane, some amount of the cholesterol part is also there. This is also the one type of the molecules, cholesterol. Microtubules, some are the attachment at the outside. Then glycolipid molecules, we are say this is the glycolipids molecules. So I hope you will understand now the fluid mosaic, this fluid mosaic model better way. So this is a two layer of this lipids, abundant protein, peripheral protein. This is important, and here this part is called carbohydrate. Okay, we are saying there is some amount of the carbohydrate is also present. That is a polysaccharide base. That's why it is a chain form you will see in this structure. Fluid Mosaic model, there is some remember points. So see, proposed by Singer Jonathan, okay, as Jonathan Singer and Gert Nicholson. It states that the plasma membrane is formed of protein iceberg in a sea of lipids. Plasma membrane is lipoproteinous and trilaminar. Okay, lipoproteinous and trilaminar. So see how it is. Phospholipids bilayer is fluidy in the nature. Okay, as I say that lipids are having a eyes related, very similar in the eyes. So fluidy in the nature, in which phospholipid molecules show the two type of movements. The one movement, this one is you will not get in your NCERT, but your concept should be clear about the plasma membrane, because if you understand this topic, so this thing is coming in a many way. So two types of movement, transition movement and flip-flop movement. Transition movement, a molecule change their position within the same layer. Clear? Flip-flop movement, molecule changes their two layer can be interchanged, that is going to flip-flop, interchange, one is out, one is going to in. So flip-flop, so that one is lipids, one related description. Now the protein. Proteins are globular and are of two types, extrinsic and intrinsic, as you see in that diagram also. Extrinsic protein is also called peripheral protein. Intrinsic protein is also called integral protein. See, phospholipids molecule is a polar molecule, has two specific end. One end is a hydrophilic and one end is hydrophobic. Hydrophilic head of a glycerol and the hydrophobic tail is a two fatty acids. So this is a head of a glycerol one. Okay, so it is a written glycerol, EROL. Okay, so just be see here. Next is carbohydrate. There is some amount of the carbohydrate is also. Carbohydrate especially oligosaccharides. Okay, oligosaccharide itself we can be say the types of the polysaccharides. Okay, where there is a two components joined to each other, are associated with the only outer surface. You can be seen that imagine also. Okay, of plasma membrane. Some amount of the cholesterol also we can be find. So cholesterol molecules are present in between the phospholipids. This is our MCQ based questions. Okay, so note down this one also. Function of plasma membrane, which is very important. It can be come in 3 to 5 marks question. It causes compartmentalization and allow exchange of material. Okay, compartment is mean chamber banata hai. Protect the cell from injury. Okay, carrier protein, as you can be see in this diagram of plasma membrane, help in active transport. Plasma membrane is this is very important point, so please just note down. So we can be say कि plasma membrane is having a semi-permeable nature. So plasma membrane is a selectively permeable or differentially permeable or semi-permeable membrane, as we had discussed the nature previous also, which regulate the exchanges of material between cytoplasm and extracellular interstitial fluid. Okay, so this is a very important helping in the exchanging of material. Now this transportation is the two-way, passive and active. If it is an energy is utilized in the form of ATP, okay, to taking off the material from here to the inside of the cell. So there is a utilizing of energy, ATP, but here is a molecule and here is a cell, but this molecule can enter inside without the utilizing of the energy or ATP. So that is called a passive process. How it is? This is just brief things you just be note down or understand. Okay, so there is a brief things you can be understand about the passive and active process, including diffusion, osmosis, passive transport, facilitated diffusion. Okay, as I said, there is a no utilization of energy in movement of material from out to in. Okay, so diffusion along concentration gradient, it means higher concentration to the lower concentration, the movement we will see in our body. Pulmonary gas exchange, it means through the lungs. Osmosis, passive movement of water or other solvent. Okay, so we see this thing in osmosis. Osmosis itself is a water movement. So selective reabsorption of water and urea from nephric filtrate. Passive transport, material moving through the channel in carrier protein along the concentration gradient. Okay, you can imagine the series are protein, the series are layers protein, and here the material. This material is taken by this carrier protein and going inside in the cell one. So material moving through channel in carrier protein, this is called a carrier protein also. Okay, it's carry that specific substance or the molecule along concentration gradient. So we will see in our body in that nerve induction process, it means inward movement of sodium. Okay, sodium is just move inside. You will be seeing this one in chapter number 20. Facilitated diffusion along the concentration gradient. This one is not against, along, because here is no utilizing of energy, that's why. So along concentration gradient but assisted by carrier protein. And here the absorption of the fructose and entry of a glucose in RBC. These things we are going to see. So this is a passive process. Active process is having the types: active transport, endocytosis, exocytosis. Active transports, it means energy development movement of material. Okay, energy dependent movement of material against concentration gradient assisted by carrier or permease. This is our enzyme name. So what we can be say, the against the concentration, so they have to use the energy to the movement. So that's why active transport. Endo, it's inside, cyto is in the cell. If the materials go inside from out to in, that is called endocytosis. So intaking of material by cell through the cell membrane is a utilizing energy, is energy dependent. So this one is also is a active process, endocytosis. Exocytosis, expelling, it means substance or the molecules go out from cell to the outside one. So expelling of material out of cell through the cell membrane. Example, release of secretion from gland cells. Okay, gland cells, you can be say digestive gland is there, salivary gland is producing saliva. So this one is a releasing. So it is also used energy. So that's why active process. Endocytosis is also the two-way. Okay, what is that? Pinocytosis and phagocytosis. Pinocytosis, it means a liquid related. So intaking of a large size liquid nutrient. Okay, if it is a nutrient is a liquid way, but that is having the big size, so they are utilizing the energy. We see intaking of iron by bone marrow. Okay, and intaking of muscles is a fat molecule by intestinal cell. We see this type of pinocytosis which is utilizing energy. Phagocytosis. Phagocytosis, intaking of large size solid particle, okay, engulfing of microbes by phagocytosis. Example, ingestion in amoeba. Okay students.